The last two lectures, we just started aerobic respiration and we completed glycolysis and the citric acid cycle. Now we're on to stage 3, which involves electron transport chain and oxidative phosphorylation. So if you remember from the first two stages, we produced only small amounts of ATP with a lot of electron energy that's being carried by coenzymes called NADH and FADH2. These electrons, these high energy electrons, will now be used in stage 3 to actually produce a ton more ATP through electron transport and ultimately production of a hydrogen ion concentration gradient and the use of a protein called ATP synthase. So let's start and discuss this stage. So few amount of ATP is actually produced in the first two stages. So where do we get it? And now let's talk about the process. This is just showing you an overview of everything we've talked about so far. The gray box is representative of a mitochondria, and at the end product, pyruvate from glycolysis is transported directly into the mitochondria and converted to acetyl-CoA. Acetyl-CoA is then picked up by the starting enzyme of the citric acid cycle and ultimately oxidized in this pathway, which results in CO2 production. These oxidations and rearrangement steps result in production of energy, which some is in the form of electrons. Electrons from this stage, as well as glycolysis, are now going to be filtered into stage 3, the oxidative phosphorylation process. The stage 3 is described as what's called chemiosmosis or chemiosmotic coupling. It involves the production of a hydrogen ion concentration gradient. This energy of the concentration gradient will then be utilized to make a lot of ATP through ATP synthase. So we're coupling two different processes, a hydrogen ion concentration gradient to ATP production. The electron transport chain that's involved in moving electrons and thus generating a hydrogen ion concentration gradient in the intramembrane space is found on the inner membrane of mitochondria, and we have many different electron transport chains and associated ATP synthase lining all over our inner mitochondrial membrane. You can see these three important pumps which are shown in this diagram, and the adjacent ATP synthase, which is going to be responsible for ATP production. So two important questions arise when we discuss the stage 3 stage. Where does the electron transport chain get its electrons? And if the electron transport is pumping ions against a gradient, where does it get its energy? And the answer to that is both from NADH. And the important material that NADH is transporting are the electrons. The electrons provide the high energy for pumping mechanisms that allow for the hydrogen ion concentration gradient to ultimately be produced. And NADH obviously is responsible for delivering these electrons so that electron transport can occur. Now keep in mind this is showing you the structure, the special aromatic ring that NADH contains that allows for its ability to carry electrons. This is showing you in its reduced form, and when the electrons are dropped off at the electron transport chain, it then converts back to its oxidized form so that it can be reused in glycolysis and the citric acid cycle. The electron transport chain has a distinct order of the three large pumps, which are made from many different proteins. These large protein pumps, assembled from multiple proteins, are as follows. The first pump is called NADH dehydrogenase complex. There's a movable molecule called ubiquinone that delivers electrons from NADH dehydrogenase complex to the cytochrome C complex. The cytochrome BC complex is the next hydrogen ion pump, and electrons are then moved to another movable component, which is cytochrome C. And then cytochrome C will deliver it to the final hydrogen ion complex, pumping complex called cytochrome oxidase complex. As the electrons move from the first complex all the way to the third, they lose energy. So they're at their highest energy level initially, and then they lose energy as they move through. The order of the complexes is important, and based on research in which they mixed up the order, they found that electron transport shuffling through one complex to the next only occurred in this particular order, which identified the fact that there must be a reason, and they identified that ultimately there is an increase in affinity for the next complex as it moves through, with the highest affinity for oxygen, which is the final electron acceptor, which is picked up by cytochrome oxidase. This increasing affinity allows for the movement to go from NADH, dehydroxylase complex, to ubiquinone, to cytochrome BC complex, to cytochrome C, and then last to cytochrome oxidase complex, where the final electron acceptor is oxygen, which leads to water production. If we look a little closer at the cytochrome oxidase complex, oxygen ultimately gets picked up by this complex continually as we breathe it in, and the electrons are moved through this complex due to special metal ions that reside in this complex, and this helps mediate the movement as well as the acceptance by oxygen and ultimately water production as oxygen picks up electrons. Oxygen is the final electron acceptor. If oxygen is not present for aerobic respiration, then the last two stages of aerobic respiration will not occur. This would be due to the fact that if oxygen does not pick up electrons, then the electron transport chain is stuck, and therefore no additional shuttling occurs through the transport chain, allowing for pumping mechanisms of hydrogen ion. This particular diagram is showing you exactly how electron movement through these pumps leads to pumping of hydrogen ion into the intermembrane space. Due to movement of electrons, it leads to different conformational states of the pump, allowing for release of hydrogen ion into the intermembrane space. This movement of the electrons provides the energy for these conformational state changes to allow for the pumping of hydrogen ion from the matrix into the intermembrane space so that ultimately a concentration gradient can develop. Now to look at the process overall, you can see that electrons are dropped off at the first complex, at the NADH dehydrogenase, are then shuffled by ubiquinone to the next, which is the BC cytochrome complex, and then lastly to the cytochrome oxidase complex, where they're finally picked up by water. This movement leads to pumping of hydrogen ions into the intermembrane space as those electrons move through. The more electrons we have that move through this electron transport chain, the higher concentration of hydrogen ion that develops in the intermembrane space. This hydrogen ion concentration gradient is not only a difference in concentrations from the intermembrane space to the matrix, so we've not only developed a concentration gradient, but also an electric gradient, where there's a higher positive value on the intermembrane space compared to the matrix side of the membrane. This leads to a huge driving force for the electrochemical gradient of hydrogen ion to move into the matrix through a special protein called ATP synthase, in which ATP is produced. This is a look at the structure of ATP synthase. ATP synthase has two important parts: the F0, which is shown here in red, and the F1, which is an enzymatic catalytic domain for ATP synthase. As hydrogen ions move through this protein, it causes the F0 to spin. This spinning motion leads to conformational state changes in the F0 region, leading to ATP production. It's important to note that this pump can also run in the reverse, meaning if the concentration gradient of hydrogen is eroded, where we have a higher concentration of hydrogen ion in the matrix side rather than the intermembrane space, hydrogen ion can actually be pumped into the intermembrane space, which requires ATP hydrolysis. So it requires an energy source to do so. This is thought to be due to the fact that mitochondria were originally a bacterium that was engulfed by a primitive eukaryotic cell, and this might have been a hydrogen ion pump that was important in developing hydrogen ion concentration gradients outside the bacterium, allowing for something like co-transport of nutrients to occur for the bacterium. Obviously, this is the direction in which we want the motor to run in eukaryotic cells so that we can ultimately utilize it to make a high level of ATP in the last stage of aerobic respiration. So as review, in glycolysis and citric acid cycle, we ultimately made approximately 4 molecules of ATP/GTP. At this final stage, with the complete oxidation of glucose, the energy and electrons that are released are utilized to make between 32 and 34 ATP in this final step, which is essentially the highest energy yielding step of aerobic respiration. So I have an assignment for you to complete for at the end of this process. One is I want you to draw take a piece of paper and draw a large diagram of a cell with a large mitochondrion inside, and I want you to break down in the steps that we discussed of glycolysis, conversion or the end products of glycolysis, where does it go, and the filtering into the citric acid cycle, as well as describe the electron transport and the electron transport chain and ATP synthase. So do a diagram of this process, and then on a separate page, I want you to write in your own words a brief summary of these steps of all 3 stages.